Hi everyone, welcome to Hari's Success Point. Today in this session, we will discuss the 10th chapter of Unit 3 of 11th class biology, that is cell cycle and cell reproduction. As you all know, living organisms have the inherent ability to grow and reproduce. Growth and reproduction occur at cellular and organismal levels. Cellular growth is accomplished by the intake of substances and the intracellular synthesis of biomolecules. Cellular reproduction occurs by the production of new cells from pre-existing parent cells. It involves the synthesis of cell constituents, duplication of chromosomes, nucleus, and cell organelles such as Golgi bodies, centrioles, mitochondria, microbodies, and chloroplast, and the division of cells. A growing and dividing cell passes through a definite cycle of events known as cell cycle. Cell cycle represents the orderly and cyclic sequence of events or phases through which a dividing cell passes from one division to the next. In it, the cellular material gets doubled or duplicated and almost equally distributed between the daughter cells. The length of the cell cycle varies in different kinds of cells. A standard or typical eukaryotic cell cycle consists of 4 non-overlapping linear phases, namely G1, S, G2 and M. G1 and G2 are the first and second gap phases during which DNA synthesis does not occur. S is the synthetic phase characterized by active DNA synthesis. M is the mitotic phase or division phase during which cell division occurs. G1, G2 and S collectively represent the non-dividing phase of the cell cycle, often called interphase. The switching over of the cell from one phase to the cell cycle to the next is called cell cycle transition or phase transition. After division, a proliferating cell will enter the G1 phase of the next cycle. Often, animal cells exist from the cell cycle after division and enter a non-proliferating resting state called quiescence or G0 phase. So the cell cycle begins from the G1 phase. G1 is the first gap phase between the end of M phase and the beginning of the next S phase. During this phase, the cell is very active in the synthesis of RNAs, proteins, carbohydrates and lipids, and it undergoes metabolic preparations for the synthesis of DNA in the S phase. The DNA content of the cell will have 2C value, that is double the value, and the chromosomes will be single stranded during G1 phase. Then comes the S phase. This is the synthetic phase between the two gap periods. It is characterized by DNA replication, chromosome duplication, synthesis of mRNA, histones and proteins, and the organization of nucleosomes. Thus, it is at this stage that the quantity of the cell's hereditary material gets doubled. That means the DNA of the cell doubles. During this stage, chromosomes will be double-stranded and the cell will contain double the amount of DNA. So, during the G1 phase, the cell was having 2C value of DNA, and when it comes to S phase, the amount of DNA will be 4C. That means double the value of DNA compared to the DNA content in the G1 phase. Then the next phase is G2 phase. This is the second gap phase between the termination of DNA synthesis and the beginning of the cell division. It is characterized by the continued synthesis of RNAs and proteins and the duplication of cell organelles. During this stage, the nucleus attains its maximum size and the DNA content of the cell will have 4C value. That means twice the amount of the DNA of the G1 period. Some proteins synthesized during this phase cause the condensation of chromosomes for cell division. Then comes the division phase, that is M phase. This is the division phase characterized by the movement of centrioles, appearance of mitotic spindle, separation of the sister chromatids of each chromosome, migration of daughter chromosome to opposite poles, nuclear duplication, cytoplasmic cleavage, and cell division. The DNA content in the daughter cell will have 2C value, that is half the amount of DNA in the G2 phase. In M phase, two types of divisions can be seen. They are mitotic division and meiotic division. In this session, we will be discussing the mitotic division that is found in cells. Mitosis is the characteristic type of cell division which keeps up the genetic continuity of organisms. In unicellular organisms, it serves as the fundamental mechanism of asexual reproduction and genetic transmission. But in multicellular organism, it is a means of growth, development, repair, and regeneration. The mitotic phase is conventionally divided into four major stages, namely prophase, metaphase, anaphase, and telophase. The major cellular events occurring during each stage of the cell cycle are different. The first phase of the cell cycle or mitotic cycle is the prophase. Prophase marks the onset of mitotic cycle. It involves both nuclear and cytoplasmic events. Nuclear events, more specifically chromosomal events, include spiralization or coiling and condensation or shortening of chromatin filaments to form short and thick chromosome. Then polarization or outward movement of chromosomes towards the periphery of the nucleus, and the longitudinal splitting of each chromosome and the formation of kinetochore. The cytoplasmic events include the formation of the mitotic apparatus. Then comes the metaphase of the mitotic cycle. In between the prophase and metaphase, there is another intermediate phase called prometaphase. This stage is marked by the dissolution and disappearance of nucleolus, disintegration of nuclear envelope, disorganization of the nuclear pore complex, and the depolymerization of nuclear lamina. Metaphase is initiated by the movement of chromosomes towards the center of the cell. This centripetal movement of the chromosomes is called metakinesis or congregation. In the center of the cell, the chromosomes get attached to the chromosomal spindle and arrange themselves in a single plane or row in the equator of the cell. This is called orientation. The equatorial array of chromosomes formed by orientation is called equatorial plate or metaphasic plate. The chromosomes attach to the chromosomal spindle with the help of their kinetochore. The sister centromeres of each chromosome attach to the spindle from opposite poles. This enables the separation, segregation, and migration of the sister chromatids to opposite poles during anaphase. The third phase, that is the anaphase, begins with the separation of the sister centromeres and the segregation of sister chromatids from each other. All chromosomes of the metaphasic plate split almost simultaneously. Soon, the sister chromatids move to their respective poles at a uniform velocity. Now they are called the daughter chromosomes. The last phase of the mitotic cycle is the telophase. The end of the migration of chromosome towards the poles marks the onset of telophase. At its beginning, the chromosomes are tightly coiled and they remain in two groups at the opposite poles of the cell. As far as cellular events are concerned, telophase represents the prophase in reverse or the reversed prophase. That means all the things that happened in prophase occurs in telophase in reverse direction. During it, the cell returns to its interphasic condition. The major events of telophase include despiralization, condensation and elongation of chromosomes, reconstitution of nuclear envelope, reappearance of nucleolus and reconstruction of nucleus, dissolution of mitotic spindle and cytoplasmic cleavage. So, a cleavage in animal cell, a cleavage furrow appears in the plasma membrane around the equator of the cell, and thus the cell divides into two. In most plant cells, cytokinesis or cell division occurs by cell plate formation. In this case, a barrel-shaped region or body appears in the equator of the dividing cell. These vesicles fuse together and form a semi-fluid plate called cell plate. This plate grows sideways, touches the side walls of the cell, and forms a complete partition inside the cell called middle lamella. It divides the parent cell into two daughter cells. The cells on its sides will synthesize and deposit cellulose, hemicelluloses, and other substances to form a primary cell wall on either side of it.